Okay, thank you for being here in person. Let me see. Okay, I recorded this. So I just assign every of you for the next homework is to explain dot product, geometric meaning of this dot product, and also applications, and also explain PCA. Try your best to take a small video to explain this. Your target audience are the people who don't know this, and you can use your plain English to explain them, the concept to them. What I found recently, this PowerPoint has this recording function, and it's pretty neat. And you can have your slide, and also at the same time when you present, record everything. So I think this is a new function. I just saw it yesterday. So what we are going to do today is I will lead you to do this numeric experiment to start again from very beginning, the dot product of two vectors. And then we will jump to examples of PCA. So we will use some example data set and also the T-part. Okay, we have T-part. Then we let you to try use PCA to real single-cell RNA-seq expression matrix. Then we will look at tSNE, tSNE method. It's also built-in function in MATLAB. I will show you how to find the function of UMAP. UMAP is not a built-in function yet in statistics department toolbox, but their online community, someone developed this UMAP algorithm in MATLAB. Otherwise, you can use R or Python to run UMAP or other these methods. I shared with you through Yongjian sent out this paper. It's recent paper talking about the dimension reduction technique. And the idea of this paper is to synthesize different methods. You use tSNE, you use PCA, you use UMAP, whatever. You have a lot of them, and you do a math synthesis of all these coordinates and come up with a new solution. And this new solution, according to the authors, is a better solution than any individual dimension reduction method, because it takes all the advantage, which consensus information across all these methods results. The paper I just want to show you, this is an active research field. People started still working on different one. And I believe there's a new one will coming as well. There's another group is developing a new dimension reduction algorithm and for single cell data specifically. So I probably will send you another paper to see. Okay, so so every year there's some small changes. So the PCA is basic, the tSNE UMAP is relatively new, and we have new methods coming up. So for this new method, if you have most of them implement in R or Python. So today I'm going to show you if we have time. So if you have an R package, can you use it in MATLAB here? So through the interface, I show you, okay, if you set it up, you can directly use to call R package to get the results and put in here. Or either way, if you want to use the output from the MATLAB, this environment, and save to R format file, you can still have the communication. If you're familiar with R, it's fine. This is open framework. Yeah, let's start from this. Okay. So this is a start page. So I want you to remember this. Two vector, if you do dot product, it means that it's the norm, the length of the first vector times the length of second vector times a value which is cosine between its angle. So now we are going to set B have a length equal to 1. So you normalize, you make a unitary vector. So when one of these, either V or U, you use as a new axis and make this norm to 1, then the dot product between of these two become one of these length times the cosine theta, what is the projection of one vector onto the other. So that's a new coordinate basically. So we are going to try one of this example in MATLAB. So we don't need a toolbox. We just come up with a say A is a 0.2, 0.3. Okay, make it a vector, so we make it vertical. Is this a unitary vector? It is not, because the length of this norm is not 1. Okay, so we make it a unitary vector. We have to do a divided by this. All right. What I made is this vector is too short. It's not a unitary vector. Why I want to make it a unitary vector? Because if I multiplicate this to another vector, I will let another vector to project onto this one, but not change the other vector's length. So you can use the dot product directly as the new coordinate as A when A as a x-axis. I hope you are with me. Let me do that again. Okay, good. So that's my old xy axis. My 0.2, 0.3 is here. Okay, so that's my random number I just created. However, as norm is not 1, it's too short. So I make it unit vector and make unitary vector 0.55, 0.83. But I just rescale it, right? So that's my A. Is that right? Okay, everybody. Right. Now, if I have a point, say 2, let's see, whatever, I make it up. So somewhere here, a B. So we have a B here. Let's see, 0.3. 1.2, something like that. Okay. So I'm going to use A times B, B times A should be the same, right? Because the dot product is a scalar. So let me see. So 1 become, so this is dot product, right? Or you can write A transpose times B, the same. Dot product is a scalar. So what that means is the 1.6. So you project B onto A. So its value 1.1649 is the length of here. That's 1.1649. Okay. So in this case, you can consider A as your new axis of X. So your B, it was based on the original coordinates, but now it has a new X axis value, which is 1.649. Okay. So that's a meaning. So you actually trade A vector as a new axis to allow B to have a new value and based on the A as a new axis. Now I'm going to create A is this. Instead of do A, I'm going to create, I still call it A, but it's a set. A first minus A second, A second, A first. I manually create this two vector sets. Okay. So now my big A contains two vectors. One is this original 0.5, 0.8. Another is negative 0.8 and 0.5. Negative, so it's if you draw it correctly, it should be okay. So that's my A, another A in the set. Okay. So artificially, I create this to make the set. So this set, this A contains two vectors. They are orthogonal to each other, and also they're all unitary vectors. And the norm of A vector norm of A, they all 1. Okay, so I make this A and small a primer. So now I can use A to rotate the B directly. Okay, the big A. Yeah, so let's see. Something wrong with A. Let me see why. Okay, so this is the first, this x-axis times b. Well, I have to do dot product. So I have to transpose this. So this is what we did first. So if I treat the first column as my x-axis, I get this. If I treat my second, I get the projection of 0.41. So 0.41 is the B project onto this axis. Okay. So the length of this is 0.41. 416. Is that okay? So if I have this A as a set of new axis system and have this vector and that vector, so if I multiplicate A as a whole with B to do the dot product, I get the B's new values for B's x axis value and y axis value. No, still doesn't make sense. So now this big A contains two vectors. And the first vector is this, okay? Is this. Second vector is this. So I'm using big A, which combine small a vector and another as a set of my xy axis, the new system. So when I multiplicate A transpose with B, what I did is I multiplicate B with A and A prime individually, two dot product operation. And when I multiplicate B with a small a here, and I get the length of this part. When I multiplicate B with A primer, I got the length of this part. So I can use these two lengths as my B's new coordinate value. When I trade this A as my new axis, axis and A primer as my Y axis. In that sense, you use this matrix to rotate B. Okay, you rotate B basically. The B was value have this and that. Now you have B value as this and that. It's so confusing. So the projection basically it's let's do that. How can I change? Let's make a new one. Okay, my A. So that's my old system, and then I define first vector 0.5, 0.8. Somewhere here, okay? So that's my small a. Then I gave A and another A, okay? This is this one. And make them, force them to be orthogonal. Okay, this is my old x, y. But now I'm going to use this as my new x, and that's my new y. All right. So what I can do, so once I have this system, so what I can do for a point B here. So this point B had the value as for their original x, y. You have this value and that value, right? That's x, y. But now I'm going to compute the B's coordinate value according to for my new axis y. So what I can do is to project B onto this axis to compute its new distance of this, compute its distance on project onto A, and then compute its distance project onto A primer. So all I have to do is to know these two distance, d1, d2. This d1, d2 are two values, but these two values, d1, d2, once you give to B, is B's new coordinate value, which use this A and A primer as new x and y axis. So the easy way to compute this is you put A and A primer together, make this big A, and then you do the dot product for A dot B, you get these two values, d1, d2. Okay. So that's what I did. I have have to do the transpose in order to, and I get the new coordinates. So B was 0.3, 1.2, but after you use that as a new coordinate system, then you have a, once you compute this, you get this. Okay. When you have a lot of B, you can do the same. This is just one point, but if you have a B contains a large number of points, you do just one operation like this, you get all the new coordinates system changed. Yeah, you have to, maybe you have to use some of these examples to figure it out yourself. Yeah. But that's exactly the meaning of this projection, okay? Projection. Okay, what I showed you is just rotate just one data point. If you have 3 data points, for example, example like here, okay? Also, this is, I made it up. You can see, okay, I have 3 data points and centralized, and you can type this in if you like. And then see, I want to use E, this vector, as my new x-axis. So what is the value for these 3 data points, x-axis value on here? So you can give it a try, and I will type that in. So I have a data, 3 data points now instead of 1. So I have data equals 2 minus 2.2 58, 0.3188. You can actually draw these 3 data points on the on your paper. To centralize them, I can you can do minus its mean. Okay. Okay, this mean operation gave you the average by rows. Okay. For this data set, first row is their x-axis value, and second row is their y-axis value. When you do this, you move your origin of this point 0, 0 into the middle of these 3 data points. Okay. That means centralized. If you think about where they are, and there's -2, 0.6, they're not centralized, but after you do this, they distribute it around 0, 0. And when you take a look at their average of their mean is in the center, and their y, they're in the center. So we are ready to find the rotation matrix to rotate them in the center. Okay. So this E is a new x-axis I want to use. I just random. It's a random vector to show you. Okay, you can project your old data points onto this new vectors to get their new x-axis value. You can get any E. So my small e is 0.253, 0.9596. How can you tell whether this E is unitary vector or not? I can check its norm. It's 1, it's good. So I already normalize it. So this is a unitary vector, only tells you the direction. Is okay. Now, I have my 3 data points, and if I apply E to them, let me think about this. So this is, my E is just x-axis. I don't have Y in there. So I'm going to multiplicate my E for each of these data points one by one. Basically, I will multiplicate the first one to get the dot product. What that means is I want to project my first data point onto my E and see what's the value of that projection. So my E is 1 by 2 matrix, and my V is 2 by 1. So this should work. Oh, no. My E is 2 by 1. So I have to transpose this times V the first in order to guarantee the match in inner dimension. So that's a dot product. If I don't want to spend time to figure out their dimension, I can do the dot. Okay. This always gave me the correct answer. So what this operation is, I project my first data point onto the E. What you can see from here is my first data point, V1 is here. When I project it onto E, it has some negative, the length should be, so this project onto here. I draw anything on here? Yes. This is so I project this onto here. The value I will get should be the distance of this. Okay. That's my distance, 0.4. So my second, now I'm doing the second point project on the, well, dot product should be a non-negative. Okay, for my V2, I have this 0.9 negative. So notation. So I project my V2 to here. And my distance is this. It's very small, right? It's this part. Is that very small? -9. Anything wrong with this? Let's try the third one. 0.46. Project that onto here, and then measure the distance. Okay. I can do this for not one by one, but I do this E times the whole V and directly get this 3 values. It's one operation. Okay. So the dot function doesn't work here, but this operation will give you the value. Let me see if this is correct. I think I made a mistake by taking, so I should do this times E. So it's the same thing, right? Okay, so this 3 distance, well, transpose this. So this 3 distance, what we can do is take square of this and sum together. So that will be the dot here. So this will be the squared of this distance and the sum together. So this is called the total variance of your data point project onto the new axis system, and what's the separation between them, basically. Okay. So the PCA, the purpose is try to make this as large as possible. Okay. So back to this example. So if you use this random E as your new x-axis, and you have a total difference 0.41. So this value can be improved if you change, say, okay, I try another one, it should have a larger number. Because this is a random selected vector as x-axis, not a principal component. So now your job will try a new E. Okay, just put the 2 random numbers here and then make them normalized. Unitary vector. So then you probably want to try a new E to that direction use this as axis, because that will give you the largest difference of this value. Yeah.
Is V1 or V2 the locations?
Which value?
Oh, the location of like the location of V1 is it corresponding to the second set?
Oh, so maybe this is some mistake here, right? It's not correct. So this position should be negative. Yeah, it should be here, right? Where is the? Yeah, so this illustration may be not correct. It's not corresponding to this. So I should have a better plot. This looks confusing. But you can try in paper and see, put this dot here and then try. I don't have a marker pen here. Yeah. So that's why when I compute the second, the negative 0.9 is not right. Yeah. So my first data point should be -4. So this is almost the first data point, this, right? My second one should be, this should be the second one. Yeah, so this is the second one. The third one, 0.3, 0.3. Yeah, so I mess up. So this should be the first one, and this should be the second one. So the projection of second one is negative 0.9 something. Okay. So all you have to do is to make a total of this distance. So now this is a total of distance. So I encourage you to try different E values. So if you current E value gave you this, and if you change, for example, I will change another E1, another guess, 0.65. Okay, I need to normalize it. So E1 divided by the norm of itself. So that's my second E. If I do the same and replace original E with E1, original E gave me the total sum this, my E1 may give me a larger value. Yeah, so this is much larger because the direction. So I can change and randomly guess and give me which one can give me the largest separation of the data, basically. It's all the data point to you, projection on you, and their new distance from the origin, the total sum. Okay, you probably have to go back and spend some time. I will correct those later. I think that's a good example to show you, okay, you can use this formula to calculate the total distance on new x-axis of their projection onto to 0, okay? Now we are going to look at the PCA. So PCA will help you to find the best this coordinate system to separate your data, okay? So we can use example they provided in PCA. So it's very easy. And if you gave it the data matrix X, PCA function will return a principal component coefficient. Okay. So your big X is n by p data matrix. Remember, in our vector-based data points, their samples are in the column, but here is transposed. So now the samples, you have n samples, and the p are the variables to define those samples. So it's very different from, it's different, just opposite from transposed from what we have in single cell data. We treat rows as genes, columns as cells. So when you do PCA, you need to first transpose your UMI matrix to make sure, okay, the PCA apply to rows for the samples. Otherwise, you're doing a PCA for genes. So once you gave the data, and the PCA will look at, okay, there are P variables. Basically, that's a dimension of your data. Okay, you use how many genes to describe your cell. And then it will return a P by P matrix, which is a rotation matrix. In our previous examples, we always use 2D. So your rotation matrix is always 2 by 2, so it's very small. When you have a lot of the variables, then this P by P could be a large matrix. Let's look at their example they gave. Okay, in this example, we can first load an example data set. Let me clean my workspace. So load this. The data set is this called ingredients, which is 13 by 4. So we can see, okay, this is our data set. Ingredient. So what I did is just reassign a name for this matrix, 13 by 4. So you describe this 13 samples by 4 variables. Okay, everybody get there? Okay. If we don't rely on the PCA function, we can do centralize our data first minus its mean. Remember, we had covariance matrix C divided by 3. Let me think about it. So this I have to make it right. So is that the covariance matrix? Divided by the number of samples. So I have to divide by 13. Okay. So my covariance matrix should be a 4 by 4 matrix. Yeah. Mean. Oh, yeah. So that depends on whether your rows are the variables or samples. In previous sample, in here, in this example, my data is, so my data is in the column. My sample is a column that I have to use this. In this case, it's opposite. My samples are in the row, so I don't have to do it. So basically, this means 1. So I can ignore 1. Yeah, so I think I'm doing it right. So I want to conform with my covariance matrix computation because I compute my covariance matrix using this formula myself, but I can't use the built-in function to conform. Yes, looks the same. Okay. So I have my covariance matrix, then remember the eigenvalue. Let's see, I will give a E and a D. Eigenvalue decomposition will give you the rotation matrix. Well, so the E output is my eigenvector combinations. Is the rotation matrix I want to use. Okay, so the data set is 4 by 4 dimension. So your rotation matrix is 4 by 4. So if you can think about this like your x-axis and y-axis, z-axis, and then the first one. So this vectors tells you the direction of your new system, the coordinate system. Okay, now let's use the PCA to solve this. So call it phase PCA X. Remember, all we need is this 4 by 4 rotation matrix to tell you what's the new coordinate systems we want to use. Let's see this PCA solution and it will look similar to what we have here. There's no way I can put them side by side. Let me put coordinate system call F here. Okay, so this is from a PCA. This is from our own eigenvalue decomposition of the covariance matrix. What the difference between this is we have to reorder the columns. So the PCA built-in function gave us the best PC, which is access. We will separate your data first, mostly, is the last one. And the second PC is this. Yeah. So the direction is negative, positive, a little bit different, but the same meaning. So how can we reorder this? This should be reordered by the eigenvalues. So the largest eigenvalue is the last column. Second eigenvalue is this, and the third eigenvalue is that. So we have to reorder our columns in this eigenvector set by their eigenvalues. So the large eigenvalues indicate once you project your data onto this axis, and you get the largest separation. Okay, so there's a way to rearrange those columns. It's very easy. But I just want to show you through this numeric exercise that you build in function PCA apply to the data and give you the coordinates of the new coordinate system, and you get the same answer if you use covariance matrix eigenvalue, take eigenvalue. Okay. So the next step is to apply this. You have to rotate your data, okay? You have to apply this to rotate your X in order to get the new position for visualization purpose. Again, I have to make it right because the data is transposed. I'm not sure I can get it right. So I'm not going to do it myself, but I say, okay, I'm going to ask, please apply this rotation matrix to my data to give me the scores of my new positions. Okay, so the scores will be the same dimension as your input. You see, score and the X are the same. The only difference is their new coordinate system that has been applied. So now you can plot the first two coordinates to separate the data. So for example, you can do the scatter plot, use score the first dimension, score the second dimension to plot your data. Let's see if they have examples. Because they don't have examples, but it's just the way people will do. They use another example. Oh, we do have this. Okay, you have scores, and then basically that's separation of your data. Okay. Basically, that's the best angle you can find to make this most separatable. If you don't use the PC, if you do the scatter plot for the original, so X second, they're not so separable as according to not as good as this one here, basically. I don't think this is the best example because there are data points like a hairball. So it doesn't matter. You look at from different angles. But we will play with the teapot examples when we come back. 5 minutes break. Okay. Any progress? No. Oh, you haven't downloaded that. Yes. You can Google. You haven't installed the toolbox. Yeah, you Google. Bing is fun. You can see, okay, single cell GA toolbox, and you found a quick installation. Mm-hmm. SC GA, yeah. Yeah, you copy that. Yeah. Yeah, just copy and paste it. Yeah. Yeah, it should work. Yeah, you don't have the variables in there, so you can go back to the interface, export the data. Yeah, the workspace, check X. Yeah, okay. Then the PCA applied to here. That's what yeah.
It's x minus mean and x.
Yeah.
In this case.
Yeah, you can ignore this because PCA itself will internally do this centralization, so you don't have to do it. Yeah. So you can jump to next. Is that your PCA results? Yeah.
What were you expecting? Or I'm not sure.
What I can, I don't know. I don't know. Yeah. What I see, someone have a like a T-shape outcome, and it looks different from what you get here.
Okay. But in terms of the functions, it's
Yeah, it's just a PCA. Yes.
okay.
Yeah.
And these are just variables you can assign whatever, or any, or does it have to be specific?
No, this is just a variable name, so it doesn't matter.
Oh,
Yes.
okay, so it could be whatever you want.
Yeah, whatever name.
Okay.
Yeah.
Try it out again.
Yeah.
I'm not sure if this was.
You can try to plot the S0 instead of S1 and see S0. What do
Okay.
you get? Yes. Same. So yeah. So I'm going to try. I don't know the outcome of this. So I have my big X. I'm going to transpose because I know the input. Let's see, xt, okay? Transpose it. Otherwise, PCA will give you different dimension. PCA xt transpose. I don't need to do this, and I ask a score, okay? It will do the centralization for me internally, so I don't have to care. I don't need the coefficient matrix, the rotation matrix, because it will internally help me to get the rotation matrix and multiplicate to my data and give me the outcome. So that's what I want to use for the plotting. Then I can start to plot with my figure. It's a score. This is after the, okay. So that's what I get. A T shape of this. You can do 3D. 3D is you ask for the first three coordinates after this rotation. So 1, 2, 3. Then you get this. Okay. So that's my solution. Okay. I hope everybody familiar with the procedure, and then similar procedures you will get for the tSNE. You just replace this with the tSNE, but you are not going to get a score. So the first, yeah. So you can try and the tSNE and the documentation just follow. tSNE also need this transposed input. You will also get a score, and then the score you can use for 3D to see them. Because this function requires the input to be raw in the sample, raw samples. The original was cells in the column. Rows are the genes. Once you transpose it, you make the matrix input that's required for the PCA. There's some of this process you can improve the row input of X. You can do the normalization. You can take a log transformation, and you will improve a little bit about the PCA results. I'm going to do this and see what I can get the best from the PCA. Okay. So this is my row X. Okay. I'm going to norm it. Underscore norm. So this is a built-in function I had. Basically, it's doing this liberal size normalization. I will add log in there. Then I have to transform this into a PCA for the PCA. Log norm x transpose. Okay. Then I'm going to plot that again and see whether my results get improved a little bit. Not too bad, right? So this is a PCA. So instead of giving PCA function the raw data, which contains a lot of zeros, unnormalized, and some value extremely large, some extremely small. So this transformation will help you to make the library size the same for all the cells, and the log will take care of those extremely large values compressed into a scale and more friendly, basically, to PCA. So once you have this, still remember to transpose your input and then get this result. Okay. Any questions? You get this picture? I'm happy about this picture because you already see the separation of a lot of cells. Really? Oh, let me think about it. So we have 7,000. Did I do something wrong? Why I get this? I'm also okay. Yeah, so make sure this row X is genes by cell. So you have 7,000 genes and 2,000 cells. So you should have 2,000 dots here at the end. If you have 7,000 dots here, you are doing a gene PCA. Yeah, 2,000 cells. So here you should have 2,000 cells, 2,000 points here. Okay. Yeah. In to score is correct. Yes. Okay. So now we look at the tSNE's documentation. They are the same group of the dimensionality reduction. So you should have a similar structure of this input tSNE. So this will also take X and just directly give you Y. So there's no rotation whatsoever because tSNE is not a rotation matrix tool. It's just give you embedding of the Y. So let's just apply this. Same thing. If I use this transformed. Okay, I use tSNE. Okay, now I use score. See S1. Okay, same input as a PCA. You will realize this will be slower than PCA. PCA is this matrix operations, but this one you have to generate your data according to the distribution, according to their likelihood, their probability being neighbors in the high dimension, and then get this at the end. So S1, you check its dimension, it's 2000 by 2, so this X and Y, so it's 2D. So you can plot them out, but this time you have to use 2D, S1, S2. Okay. By default, tSNE gave you 2D, but you can ask for 3D. I will show you how to. See whether you can get this gingerbread. Not yet. You're waiting for. That's good. That's good. By the way, this technique is not limited to the single cell data. As long as you have an Excel spreadsheet, you have some samples of animals, maybe, and then animal have a different behavior. You have a blood pressure, whatever. You have matrix. You can apply this, right, to see. Okay, this group of mice close together as a cluster. Yeah, it's a general tool. It's not just for single cell purpose. Not two variables. You have to have a matrix. You have samples and defined by many parameters. Yeah. If you only have 2 parameters, then you don't have to do dimension reduction. 3, you can still visualize it directly. 4, maybe you want to do this reduction from 4 to 3. Maybe if you have a large one, that'll be really good for these tools. Different from what I get. Oh yes, everybody should get a slightly different, right, answer, because it's a stochastic process. Our random number are different generated. Yeah. If you do this again, you will get slightly different results. I'm glad you pay attention to that stochastic difference. So if you want to have a 3D, you may have number of dimensions, and you put a 3. Okay. Now you can plot in 3D. 1, 2, 3. Oh, no. So when you write a function at this stage, if you push the tab key and then you get the parameters, other variables, the parameters you can as input to give the algorithm. For instance, you can give the initial position of the your data. Right now it's purely random, but if you can have a PCA first, then give you PCA coordinates as initial y, it will make this algorithm easy to find the best solution because you already pre-position your cells by their PCA largely. And then this will give you a fine tuning for the position. You can ask for 2D, 3D, or even higher, okay? If you ask 3D and then you have 3 coordinates, X, Y, Z. If I ask 4, there are more. And inside, okay, I think they're doing this inside the PCA inside already. If you have a PCA components, there are other parameters. In the documentation of the tSNE function, you will find examples and you use different distance to measure your cell difference and you get very different results. By default, we are using Euclidean distance, but you can use cosine distance, you can use Hamming distance, a lot of different ways to measure the cell-cell difference. And then remember, you have to have this distance matrix, and inside the tSNE, it will compute the likelihood to be neighbors, how likely they're close to each other. Yeah, so this is called a hyperparameters that control the behavior of your algorithm. Perplexing is yeah, so if you use different perplexity level, you get very different results. So the paper I shared with you, this combined different dimension reduction technique. Within there, you will see tSNE 1, tSNE 2. What that means is they use different parameter, hyperparameter to run tSNE 2 times, and in order to get more this kind of reduction results. Yeah, this is perplexing is fun. It's 20, 30, no difference. If you installed single cell GA toolbox, you have this function. If you don't install, you don't have that, okay? You apply this to log norm X. I think you should be able to get S2 directly. I'm not so sure, but just give it a try. Yeah, I probably made a mistake. So the output of S2 is 7,000 by 3, so it's not correct. So I shouldn't transpose this, because this function takes a matrix cell Gn by cell. So I have to take off this, rerun it. Okay, so this should be 3D by default. Okay, that's my tSNE UMAP embedding. We can vote. Do you like this embedding more than tSNE? Visually, which one is appealing? But remember, the function I make is to take this gene by cell matrix, so you don't have to transpose it. Otherwise, you get the coordinates for the genes. Okay, last, we don't have enough time to run R here, but you can give it a try. Say here, check your R environment from this two-box interface. I just want to make sure, okay, we have R installed. Here I check. Yes, I can find the program which already installed in my local computer here. So there's some communication we can do. Otherwise, you may have to install the R Studio from this THENWORKS R Studio. Okay, check your use this, check your environment. See whether you have R environment installed. If not, come to THENWORKS to install R Studio. And that's all for today. And you just prepare yourself later. Once we want to call R function, you can directly have that environment set up. Okay, I will just go around and make sure you have this R installed. Do you have that in there? Okay, good. That's good. It's already there. Great. Yeah. I don't know who installed that. It's by default already there. R is more popular. Yeah, you have that. Okay, so this is a CVM laptop, basically. Yeah. Good. Okay. Good. We are ready to go. You have R installed. Yes, good. Yeah. Maybe you also want to check Python, right? Python is installed. No Python. R, yes, for sure. How about Python? Excellent. Yeah. Not here. Okay. Python is not yet. Okay, so it's okay. We just have R installed. It's fine. Okay, see you next week. Okay, good. Keep warm.